Hello everyone, I'm Alaa, medical student at University of Khartoum, and today I will share with you presentation about the molecular characterization of complement receptor 1, Swain Langley, and McCoy polymorphism in patients with black-green mycetoma in Jazeera Mycetoma Center, Sudan between 2019 and 2020. Great thanks to all these authors who did their best to submit this paper. Dr. Alfadil Elamin, Department of Medical Parasitology, Faculty of Laboratory Medical Science, University of Jazeera, Sudan. Dr. Tahra Yahia Hassan, Department of Medical Parasitology, Faculty of Laboratory Medical Science, University of Jazeera, Sudan. Prof. Dawoud, Department of Medical Parasitology, Faculty of Laboratory Medical Science, University of Jazeera, Sudan. Dr. Azhari Mohammed Tyara, Department of Molecular Biology and Biotechnology, University of Jazeera, Sudan. Dr. Mohammed Abdelwahed, Department of Molecular Biology, National Cancer Institute, University of Jazeera, Sudan. Dr. Aliya Elamin, Department of Physiology, Faculty of Medicine, University of Jazeera, Sudan. And Faculty of Medicine, King Faisal University. Dr. Mahjoub, Makki, Faculty of Medicine, Omdurman Islamic University, Sudan. Alaa Azhari Mohammed, Medical Student, University of Khartoum. Aya Azhari Mohammed, with Medani College of Medical Science and Technology, Faculty of Medicine. Let's start. Mycetoma is a chronic granulomatous disease of skin, subcutaneous tissue, and bones that is present worldwide. It's endemic in tropical and subtropical regions. It occurs commonly in the food and was described by Gulls in Indian Madura district in 1842. The most common site of occurrence is food. 70% of cases, it's commonly seen in agricultural workers and the barefoot workers in dry and dusty areas. Infection can be caused by true fungi mycetoma in 40% of cases and by filamentous bacteria actinomycetes in 60% of cases. Repeated trauma or implantation by thorns and splinters provides a portal of entry of this organism. The objectives of this study. General objective is to identify the polymorphism in the allele distribution for gene encoding to CR1 associated with risk of mycetoma infection due to black grain. The specific objective to find out the frequency of polymorphism in complement receptor 1, to validate the PCR methods for detection of polymorphism along with the identification of the prevalence and the risk factors associated with mycetoma infection. The causative agent. Bacteria of class actinomycetes. It's classified as mycetoma or actinomycetoma, respectively. Fahal 2011, Fahal and Hassan 1992. In Sudan, the main causative agents are fungi, while bacterial agents are being predominant in Latin America. Efard et al. 2017. Materials and methods. Study design, prospective cross-sectional laboratory-based study. Study area, the study was conducted with Medani Mycetoma Hospital, Jazeera State, Sudan. Study population, mycetoma patients who attending to Mycetoma Hospital, co-patients attending to the hospital, post-sexes with different ages were included, all ages. 41 patients were included according to inclusion-exclusion criteria. Structured test questionnaire was used to collect data from study population. Genotyping for the McCoy and Swain Langley polymorphism was performed using the PCR. The finished TV uses green for adenine, red for thymine, black for guanine, and blue for cytosine. Northwest Association 2012. The bioedit. The multiple sequences of alignment of the isolates with similar nucleotide sequences obtained from the blast nucleotide will carry out to find the homology and evolutionary relation among the sequences. The results and discussion. This figure shows the electrophoresis gel of the CR1 and McCoy and Swain Langley polymorphism in 41 mycetoma patients by using one primer, 24 km, for two polymorphism, 305 base pair. This table shows the frequency of SNP A4795G and SNP A4828G in mycetoma patients. So the frequency of SNP A4795G for the valid present participants was 4 and the percentage was 50%. The frequency of SNP A4828G for valid present participant was 6 and the percentage was 75%. The frequency of SNP A4795G was not found in patients was 4, and the percentage was 50%. The frequency of SNP A4795G was not found in these participants. It was 2, and the percentage was 25%. The frequency of SNP A4795G in the total participants was 8, and the percentage was 100%. And also the frequency of SNP A428G was 8, and the percentage was 100%. This table illustrates the BGI Sanger sequences results, and here we want to verify the presence of SNP A4795G and SNP A4828G across these different primers with certain sequences. This figure shows the multiple sequence alignment of the mutant sample with similar nucleotide sequences obtained from the plus nucleotide was carried out to find the homology and evolutionary relation between these sequences. This figure shows the mutant gene. This figure shows both the protein alignment and the EK polymorphisms. And here this figure shows the multiple sequences alignment of the mutant isolate with similar nucleotide sequences that obtained from the plus nucleotide was carried out to find the homology and evolutionary relation between these sequences. This figure shows the mutant type K1590E SNP A4795G of the predicted amino acid lysine glutamic acid at position 14. The secondary protein structure of the mutant gene is shown here in figure A2, and the tertiary protein structure of the gene's mutant is shown here in figure A1. The mutant type CR1R1601G SNP A4828G of the predicted amino acid lysine glycan at position 25 is shown here in this figure. The secondary protein structure of the mutant gene is shown here in figure A1, and figure A2 shows the tertiary protein structure of the mutant genes. In conclusion, the contemporary study abstracted that mycetoma was prevalent among males and females with many mycetoma hospital with a PCR positive results and BGI Sanger sequences. The frequency of SNP A4795G shows 50% positive and 50% negative, whereas SNP A4828G shows 75% positive and 25% positive and 25% negative. The highly affected age class are found in age group ranging from 13 to 36 years. The study instituted that mycetoma infection rates are being the most dominated in males than females. The recommendation. The study recommended that extremely valid research in allele distribution for CXCR2, thrombospondin 4, and the nose sensaes. Further, nose lumbar is critically needed. Health education program for affected area should be applied. Thank you.